coming in, but I'll start now. Good evening. Welcome to the Institute on Ecosystems Distinguished Lecturer Series. Tonight we have Andrea Litt with us, and I'm going to tell you a little bit about Andrea. She's an associate professor in the ecology department at MSU. Her primary research interests include quantifying responses of animals to human activity and disturbance, exploring the mechanisms of those responses, developing tools for restoration, and informing the design of monitoring programs. She holds a BS from the University of Wisconsin, Masters of Science from the University of Florida, and a PhD from the University of Arizona. So, a pretty diverse background. And my favorite point or piece of information about Andrea is she is incredibly great colleague to have. She has a kegelator in her garage, and she's very nice about sharing it. So, one of my favorite aspects of Andrea. So yeah, the secret's out now. So Andrea's going to talk to us about bats. She's done a lot of small mammal work and has worked on the interaction of plants and animals, and so I'm very anxious to hear what she's been doing with bats. Andrea.
Thanks, Bruce, and thanks everybody for being here. I really appreciate everybody coming out on a Tuesday night to hear about bats. And I want to get to this idea about how the projects that we've been working on inform bat conservation. But I wanted to start first by sort of giving you some thoughts about why I think it's useful to go to bat for bats. But given that you guys are here on a Tuesday night, maybe that part isn't necessary. But I'm going to do it anyway, because I get excited about bats, and I've got some of my mammalogy students here who know I get excited about bats. And so the first thing I want to share with you is that bats are one of the most diverse groups of mammals on the planet. And then what I mean by that is that they represent a large number of species of mammals. They represent 20% of all mammal species. That's about 1,200 species that have been described. And you're looking at just some of the faces of some of those 1,200 species here. And these obviously have very interesting faces and nose leaves and all kinds of interesting things going on in their faces. The only group of mammals with more species are rodents. So this is a pretty interesting thing about these bats. All bats are members of the group called Chiroptera, which literally translates to hand wing. You can see way up here, this is the thumb, and all those fingers outstretched with the flight membrane between them, and then the flight membrane that goes against their bodies. And some bats actually have a flight membrane that extends between their hind legs as well. And so because they have this hand wing, they also have a unique characteristic in that they're the only mammals capable of true powered flight. So that means that they can get up and fly off of many surfaces, unlike these misnamed flying squirrels that just have a membrane between their front and hind legs, and essentially they do what you might call a controlled fall. So they have to climb up to a tall height, jump off, and then use that as more of a sail, or something like a paraglider or hang glider might do. And contrary to popular belief, perhaps, they are not flying mice. They are much more closely related to shrews instead. And so one of the things I want to do tonight is to dispel a few myths or misleading statements that I have heard over time, and in addition to sharing about how interesting they are. Bats live on all continents except for Antarctica, and they range in size from this bumblebee bat, which is about the weight of a penny, to this golden-crowned flying fox, which is not a fox, it's a bat. And these particular bats are fruit bats. They weigh about 2.5 to 3 pounds, and their wingspan is larger than I am tall. Despite being really short, their wingspan is about 6 feet. A lot of bats live in temperate places, which means they have to figure out how to make a living in changing conditions when resources might not always be available. So lots of bats will hibernate like bears. So they go find a cave or a mine where the conditions are pretty static and consistent throughout the winter, and they reduce their energy needs and rest for the winter. And then they can come out when the conditions have improved. Other bats migrate to find those conditions, and we have both kinds of bats here in Montana, some that migrate and some that hibernate. And I'll get back to the idea about hibernation here in a little while. Bats live somewhere in the range of 5 to 15 years and could live longer. We don't have great information about some of that. And they tend to give birth to just a single pup in a given year. And females can form these large maternity roosts, maternity colonies, where females go to give birth to their young, their single pup, and then they lactate and feed those young until they're large enough to fly. And these lactating females and their young form these very, very large groups, and those are really important resources for bats. Again, contrary to maybe popular belief, bats are not blind, but they do use their eyesight to see. But because they're active, or many of the species are active at low or no light, they capitalize on other senses in order to navigate much more easily in those dark conditions. So they use sound or echolocation to gain information about their environment as they move around and try to find prey. Many bats that echolocate have very high frequency calls, typically above our human hearing, and they make those calls in an extremely loud volume, to the point where some of the volumes exceed that of a rock concert. So sometimes it's good that those frequencies are above our hearing. There's quite a racket going on out there. They make use of the echoes from these calls that are very loud, and they can get very detailed information from those echoes, including the position and distance of objects, the size of those objects, and even fine detail as much as the texture of those objects. And so as a bat is navigating around their environment, they're emitting calls. And what you're seeing here is sort of time down here, and the frequency or the pitch of those calls that those bats are making. So they're emitting regular calls, waiting for those echoes to return, and then making decisions about how to move around their environment as a result. But as they hone in on insect prey, for example, they're going to emit those calls much more frequently because they need the information back more quickly. And so they go into a phase that is called here the attack phase, but we might call a feeding buzz. So they're making rapid calls so they can make rapid decisions and adjustments, and they can grab onto their prey, and then they can reduce the frequency or the rate of the calls after that. So the other way that bats are super diverse is in their diets. This is a picture of one of the maybe more infamous diets that are represented in the bat world. This is one of 3 species of vampire bats. And so these feed on blood, but again, unlike popular opinion, perhaps they don't suck that blood. They have very sharp incisors that they use to make an incision in the thing that they're trying to get their blood meal from, maybe a cow. And then they have an anticoagulant in their saliva that allows the blood to continue to flow and not clot. And then they can simply lap the blood off of the animal, and they're taking a small amount of blood, and then they fly away. These bats are in Mexico, Central and South America, and maybe get into the far south parts of Texas. Other bats feed on fruits, like this fig down here, and that means that they're incredibly important seed dispersers for those plants as they feed on them. And then still others feed on the nectar and pollen of plants, making them incredibly important pollinators. And so this is the lesser long-nosed bat, which is an important pollinator of the agave plant, which is an important pollinator, or important plant for tequila. And so the interesting thing about the tequila aspect is that the fact that bats pollinate agave doesn't really help the tequila makers. The tequila makers, they harvest the agave plants before they actually flower. So flowering agave put up a giant bolting stem. They flower once and then die. So typically, to make the tequila, they're harvesting agave plants before they flower. And so there's been some interesting partnerships between bat biologists and tequila makers to make bat-friendly tequila, where they leave a handful of those plants out so they can flower, provide food for those bats, and still have a set of plants for their tequila making. You can see in this graphic they are also important pollinators for lots of other species, like bananas and many other species. And then up here is an interesting tidbit about a bat that's very well adapted to a very long flower. And that bat has a tongue that's 1.5 times its body. It attaches way back on the sternum and extends out into a very long flower. Other bats don't rely on plants, but instead focus their diets on different species of animals. So down here, we've got a species that forages on fish, so they can detect the ripples in the water with their echolocation. They come down with their large hind claws, and then use that lower flight membrane here to scoop fish up out of the water and fly away. Other bats will feed on frogs and lizards and birds and even rodents, but the most common diet among bats are these insects up here. And that makes them important for another reason, in that they provide pest control. The little brown bat, the little brown myotis, which is one species we have here in Montana, can eat up to 1,200 mosquito-sized insects in an hour. So these are friends you want to keep around. Some people have tried to make estimates of the contribution that these bats provide in terms of pest control for crops and the lack of use of pesticides, or the increased yield that results because of the lack of insects and the lack of pesticides. And so there's a huge range of estimates that have been put out there, but they're all always quite large. So a lot of the bats, or all the bats that we have here in Montana, are nocturnal. They spend their time being active at night, and what they do during the day is they spend time in what we call a day roost. So they're finding a crack or a crevice, maybe under a bridge, maybe in a house, but maybe more typically in something like a tree cavity, in a tree trunk, maybe under the bark, or in a rock crack. And these are places where they don't have to thermoregulate very much. They can maintain their energy and not have to expend a lot of energy. They're relatively safe from predators, and they can spend the day sleeping and resting while we're being active. They'll come out at night, and the first thing they'll do is typically to find a drink, and then they go forage for food. While they're out active at night, they need to take a break and digest. And so they also find night roosts where they can stop and rest, digest for a little while before they go out and make another foraging bout. So all these different kinds of places are really important for the conservation of bats. Back to the maternity colonies of those females, the day roosts of bats during the day, so they're safe and can expend little energy. Places for them to forage, and places for them to roost at night and rest. This picture shows one of the largest urban bat colonies in the US. And maybe some of you have been here. This is the Congress Avenue Bridge in Austin, Texas. This is a place where about 1.5 million Mexican free-tail bats roost during the day under that bridge. They emerge at dusk, and they obviously create quite a spectacle, and it becomes sort of a tourist attraction for the city of Austin. And so this is a really obvious and large group of bats. But typically, they're not this obvious, and they aren't in as large a groups, which means that they're kind of hard to study. And despite all the interesting tidbits I've shared with you already about bats, we really don't know very much about a lot of the species. The International Union for the Conservation of Nature suggests that of those 1,200 species of bats, about 200, we really don't know enough about them to even say whether they're threatened to any extent. And so there's these major information gaps to know about, or to learn about bats and their natural history. What we do know about bats is that there are a lot of threats to their populations. The first being a threat that's common to lots of species, which is habitat alteration or habitat destruction. Maybe it's not as extreme as this clear cut, but it could be a change in forest structure that simply reduces the places bats can roost. Or it could be the closing of a cave or a mine that would have provided a nice maternity roost for many female bats giving birth to pups. The development of wind energy is also a major issue. Lots of bats have died through direct collisions with these wind turbines. But the other issue with wind turbines is that there's a major pressure differential as those blades turn. And so if a bat approaches that, they suffer barotrauma, which means that there's massive damage to their lungs. So they die as a result of that damage. But the good news is that there's been a lot of partnerships between wind energy, and biologists in order to reduce these mortalities. Most of the bats that die in these places are migrating tree roosting bats. And so we don't really know what is attracting these bats to these large turbines. Some have proposed that the bats mistake these turbines for trees. Others have suggested that the lights placed on these turbines really encourage insects to congregate, which then draws the bats in. But there's some conflicting evidence about those various hypotheses. But the idea with the partnerships between energy companies and biologists is that perhaps we can reduce the mortalities given what we know about bat biology. So one thing that we have figured out is that bats are more active at low wind speeds. And that's great when it comes to wind energy because it might not be productive to run the turbine at low wind speeds. So perhaps there can be some compromises made about not running the wind turbines when wind speeds are low, allowing the bats to move through, or reducing the activity of the turbines when bats are migrating in a particular area. Other solutions that have been proposed is changing the light fixtures, different kinds of lights on these turbines, so to reduce the attraction of insects and bats. So those are positive things. But the big challenge for bats in North America is a disease caused by a fungus. This is white nose syndrome. You can see the fungus growing on the noses of these bats. And we abbreviate the fungus by just PD because it's a pretty long and complicated name. And this fungus likes to grow in places that are cold and damp. And that exactly matches where these bats like to hibernate. So bats that are hibernating are seeking out conditions that are relatively uniform throughout the winter, and those tend to be places like caves and mines. And so when bats get infected by this fungus, they tend to wake up during their hibernation at a time when they're supposed to be reserving their energy stores. And so they wake up repeatedly, they essentially use up all their energy stores, and therefore they starve before spring emergence can occur. This disease has resulted in the death of millions of bats throughout mostly the eastern US. And we think it's spread from bat to bat most predominantly, but there's been a lot of efforts to reduce the potential spread by humans that are using these caves. So lots of education about disinfection of gear, boots, and clothing for people using these caves and mines. This map shows the spread of this disease across North America. So it was introduced or was first detected in New York State in 2006. The way this map is set up is the darker colors, the cooler colors, the blues and the greens, are the oldest detections. So you can see where it started to spread initially. The more recent detections are these reds and oranges, and you can see the gradual spread out from that initial detection. So as of the making of this map, the fungus or the disease has been detected in 37 states and 7 provinces. The most recent detections, you can see, are creeping ever closer to Montana, where we have not yet detected the disease. But a few years ago, there was this sort of interesting detections over in Washington, which seemingly jumped across the country, which you don't really fully understand. And then just in the last year, there's been detections of both the disease and the fungus in South Dakota and Wyoming. So that's a concern for us here in Montana. There's been lots of solutions that have been proposed and lots that are being investigated to try and reduce the effects of this disease. There's some interesting ones I've heard of recently. Oral vaccine, which would be very effective but hard to deploy. The use of UV light. So if you could spray UV light in caves, it would kill this fungus. And generally the problem with a lot of the solutions that have been found is that they're not able to be applied on a wide scale. Some of the more recent ones I've heard of are some different kinds of sprays. One would be polyethylene glycol that would be sprayed as a solution in these caves. And apparently that can trick the fungus into thinking it's drier than it actually is, so it can't grow. And my favorite is that there's a fungus on the Bolivian pineapple that can reduce the growth of different other fungi. And so the idea would be to grow that fungus, spread it in the caves, and reduce the growth of white nose, or the spread of white nose. So all of that brings me to the work that we've been doing. All that leads up to the idea that we have a lot of information needs and a lot of threats that we need to gather reliable information about. And that's where my fun cadre of people here get to come in, and we get to do really interesting work with the goal of trying to inform conservation, to provide reliable information by which researchers and biologists on the ground can make good decisions. The exciting thing about these partnerships is that they're all different kinds of scientists and researchers that are bringing lots of skills to the table. And the other thing to point out, that I have all these logos up here, is that they're partnerships and collaborations with various state and federal agencies. So biologists and researchers from all these agencies are involved, and we're also giving the information back to these agencies and the biologists that can actually use the information on the ground, which is really my favorite part. So what I want to do tonight is I want to highlight some of the work that we've been doing and just give you a taste of some of the fun we've been having to try to go to bat for bats. And I mentioned in my title that we're doing this at 3 scales. So I want to highlight 3 projects here and just give you a taste. And I want to start at the national level and the Continental Scale, and then I want to focus in on Montana and talk about some work we've been doing at the whole state level, and then finally zoom into sort of central Montana and specifically the Helena, Lewis, and Clark National Forest and some work we've been doing there. Before I do that, I mentioned earlier it's really hard to study bats, and so I want to give you guys some information about how we've been able to gain some insights about bats and some new fun technology that has really made our jobs much easier over time. And as I get into the work that we're doing, I also want to point out that I'm doing none of this by myself, and the people I highlighted and will continue to highlight through this presentation are just some of the players. There's plenty of other people that have been involved in this work that won't get mentioned this evening. And so, moving on. I want to go back to this idea about echolocation. And that's been a really important tool for us as we study bats. So I mentioned that bats, as they're moving around the environment, they have these calls that they make, that they emit, waiting for those echoes. And on this graphic, that's called the search phase. But what's important for us is that particular phase is really interesting in bats because every species has a unique call that they make during that search phase. There are different patterns of frequencies that they make. The duration of those calls differs, and the intensity of those calls, or the volume of those calls, differs from species to species. So that ability to tell species apart from their calls has been really essential in us generating some understanding about bats, which means we don't have to capture them, we don't have to handle them, and it doesn't even matter that we can't hear those calls. What we're doing is we can capitalize on technology, microphones that can pick up acoustic signals that are in the ultrasonic range, acoustic detectors that can record those sounds, and most importantly for us is software that can translate those sounds that we can't hear most often into images like this sonogram that are showing us what the bats are doing. So this is a sonogram, the picture of the sound. Down here on this axis is time. And then on this axis is the frequency that the bats are calling. So every one of those blue blobs is a series of frequencies that this bat passes through as it makes its call. And I'm going to play it for you here in a second. But before I do that, I can't help but call attention to the giant spotted bat up in the corner. Yes, this is a black bat with 3 giant white spots on it, and these ridiculously large ears that they use to pick up those echoes. This is a bat species we do have here in Montana, and the other unique thing about it is that it calls at frequencies that are just at the edge of our hearing. So I can actually play you this call without altering it, and you can hear what you're seeing here. And I'll caution you that this call I'm going to play is kind of quiet, and for some of you, if you've had any upper end of your hearing loss, you might not hear it, but we're going to try and see how it goes. So listen real carefully, and maybe I'll play it twice. You hear it?
No.
Okay, listen real carefully again. How many heard it that time? Okay, so just at the edge of our human hearing, or beyond for some. But what we typically have to do is we can't usually play those calls just as they are. Imagine back to when everybody had a record player, or if you had a tape deck that you could slow down or speed up. So when you speed up those records or those tapes, things sound like Alvin and the Chipmunks. They get real high-pitched, right? But if you slow it down, everything gets low. And that's what we do with these bat calls, is we slow them down so that we can actually hear them. So I want to play the same species call for you, but it's slowed down. So you'll definitely hear it now, and it's going to be louder. So every time you're hearing that sound, that bat is passing through all those frequencies. That's why you see that sort of swooping black or blue blob on the graphic here. And I want you to kind of remember what this looks like and remember what you heard, because now I'm going to show you the sonogram for another species that's very different. This is the silver-haired bat. This is another species we do have here in Montana. And maybe the first thing you'll notice is that it's not swooping anymore. It's just a sort of a straight line. This bat is making a call that's much more uniform in calling, in the frequency and the tone it makes. And you're seeing these changes in color here. Those are more intense sounds, so it's calling at those frequencies a little bit louder. This bat flies around in the canopy of trees, and so it can have this kind of call because of where it lives. So I'll play that one for you. This one's above our human hearing, and so I can't play I only have to play the stretched out version. You heard that one, right? Okay. So the great thing about all of these species-specific calls is that we can use microphones way up here, ultrasonic microphones, and down here we've got an acoustic detector, a recorder, that can record these calls if we place them out in the environment. And we can power those detectors with either batteries or solar panels in this case. And this set of tools and the ability to switch those calls into those sonograms and use software to automatically tell us what species are making those calls has sort of revolutionized our ability to understand bats. We still have a lot to learn, and there's still some challenges, but the software packages can recognize the characteristics of different species, and it can use algorithms. That's not a bat. Could be. It uses those characteristics to distinguish species and saves us some time, because it means we don't have to be out there at night catching bats all the time. We can put these detectors out, we can walk away, and then come back later, and then we have a lot of data on our hands. And so every one of the projects I'm going to highlight tonight uses this acoustic information, and I'll get into the details a little bit more when I get into this, as some aspect of the work that we're doing. So again, I want to start out at the continental or national scale, and I want to highlight that this is, again, a big partnership. For doing this work, I was collaborating with Katie Banner, who is here in the audience. While she was doing this work, she was a postdoc, but now she's on the faculty in the mathematics department here at MSU. And also Kathy Irvin, who's also here, who's a statistician with the U.S. Geological Survey. And we are fortunate enough to get to partner with another group, which is the North American Bat Monitoring Program, or NABAT for short. Notice the interesting logo with the 3 bats, which I think is very, very cool. The goal of NABAT is to use these acoustic data to try to help detect changes in where bat species occur and in the size of their populations. So the goal is to have reliable information that can then be used to make decisions. And I've said declines up here, but that's just because that's how things have been going for bats. Let's hope that at some point we're detecting increases in bats, in bat populations in places where white nose has been really detrimental. And so the goal for us is really to answer this question of how can we detect declines or changes reliably. It really is a question of information. How much information do we need to gather to be able to tell when changes are occurring? So for example, let's take the example of the big brown bat, which is pictured here. And let's say we want to know where in the northeastern quarter of the country this bat species occurs. What we could do is we could use those acoustic detectors, we could put them in places on that landscape, and we could see if we record them or not. Once we got that information back, we'd have some places where that species occurred and some places where they didn't. But if you imagine this like a puzzle, the yellow spots are the only places we have any puzzle pieces. So that means there's a lot of information we're lacking. We wouldn't know what's going on in between. So what we do is we make predictions. We make predictions about the spaces in between. So here, I'm showing you there's 30 spots on this map up here, which is places where we collected information. And we make predictions about places where we're likely to find that bat species, or places where we aren't. So green in this map is places where we have a high chance of finding that bat species, and the red are places where we have a low chance. And we make those predictions based on characteristics at every one of those detectors. If we find that bat in a place that has a lot of forest cover, with a certain type of dominant tree, and maybe at a certain elevation, then we can reasonably assume that we could find that bat species in other places that look like that. And we can do the opposite for places where that bat doesn't occur. But again, when we only have sort of 30 spots of information, 30 puzzle pieces, we're probably not very confident in those predictions. We're making lots of guesses. So we can also make maps that show how sure we are about those predictions. And now this map down here, green is not great. Green means we have a lot of uncertainty, and that is kind of across this whole board. So in this case, we've expended relatively little effort in terms of sampling, only 30 detectors, which seems a little low given this large area. So if we increase our effort, we put 273 of those detectors out, so lots more black spots. We get our predictions. Now we feel a little bit better about those predictions. We have more certainty about those, so we have lower uncertainty, and we feel better about those predictions. Making guesses between those locations, educated guesses, I'll say. But all this is really a trade-off. It means more people, more equipment, more resources, and definitely more money. And so what we're doing as part of this project is providing some different scenarios for the land management agencies and the biologists that will go out and do these surveys to say, if you have this level of resources, this is the predictions and the uncertainty that come with that. So you have to think about these as trade-offs. More information is always better, but it's always more costly and more time-consuming and requires more people. The other aspect of this work that requires a lot of time and people and energy is dealing with the recalls when we bring them back from the field. And so I mentioned earlier that we can use software to auto-identify the species that make the calls. But the software is imperfect. It's an algorithm. It's looking at characteristics that are present based on our knowledge of bats. And so sometimes the software gets it wrong. And sometimes the software gets confused about two species that might have pretty similar calls. The nice thing, I guess, in these circumstances is that people can detect patterns very well, can detect visual patterns very well. So the biologists or the data analysts are often going in and doing a bunch of hand verification. They're checking to see if the software program is correct, which is great, but it's a lot of time and a lot of money and people power. So one of the other things that we did as part of this work was to try and provide some guidance to the biologists to say, how many of those calls do you really need to look at? And are there particular kinds of calls that maybe you would focus on to reduce the effort? And with all this work, what we're really trying to do is try to allow agencies to use their resources most efficiently and get the best data. From here, I want to zoom into the state of Montana. And again, this is a series of partnerships to do this work. And in this case, Wilson Wright, who's a research scientist here at MSU, present in the audience. Again, Kathy Irvin from USGS. and Emily Almberg, who's a disease ecologist with Montana Fish, Wildlife, and Parks. And what we wanted to know here is that given that we haven't detected white nose syndrome in this state yet, how do we make sure we do once it arrives? And in Montana, we've got 15 different species of bats, and many of them are susceptible to white nose syndrome. And so the question is, how do we go do our sampling and survey effort to make sure that we can detect the disease that's going to affect a lot of our species? Some of these species are carriers of the disease, and others will be affected directly. The way we detect white nose is we capture bats, and we look at their bodies for evidence of that fungus. Up here, this is evidence of the fungus that you can see under a black light. So this is another aspect of damage that this disease does, in addition to the fact that they wake up from hibernation. So to detect white nose syndrome, we would do lots of bat captures. We'd handle lots of bats, and we'd either observe their bodies for the fungus or for white nose, or we would swab their bodies to tell whether the fungus was present. But again, resources are always limited. Montana is a very large state, and so where do we direct those energies to best tell when this disease has arrived? In other parts of the country, the ideal place to do this is in those winter hibernacula, in those caves and mines where there's large aggregations of bats. So you might go there in the spring, just before the bats are getting ready to come out of hibernation, and you could catch bats and have a lot of individuals that you could test. But in Montana, we don't know about very many hibernacula. Every one of these red spots is a known hibernation location of bats in Montana, and the size of the dot is proportional to the number of bats that were recorded in that hibernacula. So the other thing you'll notice is that there's a lot of small dots, and those small dots correspond to groups of bats that are less than 100, maybe even less than 50. So this is not a great strategy for us in Montana, not to mention that there's huge parts of the state where we don't know about any hibernacula. So if this were the first place for the disease to arrive, we wouldn't pick it up. So we took another strategy. We capitalized on what we learned at that national scale, and we built more maps of where we were most likely to detect bat species, specifically the ones likely to be affected by white nose. So in this case, we want to focus on places where we have a high chance of finding that bat species. And ideally, that would also be a place where we feel pretty good about our predictions. So we can use those same kind of maps to prioritize our sampling. And then we brought in some additional information that we had to further address this question. This is showing the data, the actual number of bat calls. So bat calls per night over here, and time over here, so days across the year. So this is about May, and then all the way to the end of the calendar year. And this is data from a single detector just outside of Helena. And what you're seeing is bats are not very active over here. Come May, we've got somewhere in the vicinity of 5,000 bat calls per night. And what that tells us is not just when bats are active, but as we get more bat calls, we think that might tell us something about bats being more active, which might have some relationship to how many bats are present. So ideally, we'd go to the places where we're likely to detect the species. We would look at places or times when we're pretty sure there's going to be a lot of bats there, and that would help us prioritize our sampling. So for every one of these dots up here on this map, we've got 1 to 3 years of data that look like this. And so this is all that same detector. And now we have a sense of how consistent that activity is over multiple years, and it tells us where we should prioritize our sampling and maybe even when. So the cool thing about all this is we built an app. We built a desktop app that our biologists can use to bring all this information together and help them plan their survey efforts. So from their desks, as they're getting ready to think about their winter sampling, they can look at places where bats are likely to be detected, and also overlay that activity data to figure out where they're most likely to catch bats. And again, the goal here being to allow the agencies to be most efficient with the resources they have to make good decisions. Okay, and finally, zooming in to Montana, specifically the Helena, Lewis and Clark National Forest. This is work of Shannon Hilties. She's a master's student in the ecology department. She just finished her second field season and is working towards earning her master's. And this work is supported by a whole host of different agencies that have funded it. And what we're interested in now is more about the habitat question. Instead of about disease or these large-scale changes, we want to know how changes in the forest are affecting our bats in Montana. And specifically, we're focusing on this little guy. This is the mountain pine beetle. This is a bark beetle that is native to the western US. It lays its eggs under the bark of pine trees, of pine species. While it's under there, it reduces the water flow and nutrient uptake of the trees. And you guys probably have seen this. The needles turn red, they fall off, the tree starts to die and eventually falls over. And this would be a typical disturbance in our western forests. However, as I'm sure many of you have seen, these outbreaks are now much more extensive than they would have been historically. We think this could be because of climate change that's allowing the beetles to persist for longer periods of time, or very dense forests that allow the beetles to move more readily and infect tree to tree. So you see a sort of sea of red followed by the sea of gray, and then eventually you'll see some gaps in those as those trees fall over. What we don't know is how this will affect bats. Other researchers have looked at how this disturbance affects things like woodpeckers, because the woodpeckers can live in the cavities of the trees as they die. Or thinking about insects that might be able to be more abundant when there's growth of vegetation. But we don't really know how it's going to affect our bats. So this map shows you the extent of mountain pine beetle damage in and around Helena. So Helena here, and so the Helena, Lewis, and Clark National Forest is an extremely large set of forest compartments. And you can see the red is the most severe mountain pine beetle disturbance. The yellow is a moderate severity, and the green is a low severity. So large areas affected, and we have the whole range of severity spectrum here. So we thought this would be a good place to tackle this question that hadn't been tackled for bats. So again, with other species, the dying trees provide lots of habitat. And we thought that actually could be good for bats as well. Bats could use these cavities in trees or roost under the bark during the day, and that really could be beneficial. In addition, these openings and increased growth of the understory when the sun permeates the forest floor could allow for more insects that also could be good for bats. So we want to tackle both of those pieces with this project. So we're again using acoustic detectors to get at this idea about foraging. If we put these detectors out in different types of forest, in different places with varying severity of mountain pine beetle, we want to know, does it change how much foraging activity, how much feeding these bats are doing? And so we go back to this idea about echolocation to do that. And instead of focusing here on those calls that are characteristic for different species, we're now focusing on these things we call feeding buzzes. We want to know, are there more of these feeding buzzes in some places than compared to others? And that's what we're doing with the acoustic data. To get at the idea about roosting, we're actually catching bats. And to do that, we use these very fine mesh nets. Maybe you can see it here. The bats typically can't detect them. We stretch them out over places where they're likely to be active. They fly through. Often we're stretching them over water. So we're picking up when they're coming out to get a drink or coming to forage. And then we put these tiny little radio transmitters on them that weigh just a fraction of a gram, so less than a quarter of a gram. They have an antenna on them, and they send out a signal, which then we can track. So at night we have to catch the bats, and then during the day we have to track them. So Shannon doesn't get a lot of sleep. And she tracks these bats to places like this. So she tracked one of her bats to this tree, and it was under some of the sloughing bark. But what's interesting about the work that Shannon's been doing, she's still doing a lot of the data analysis, so there will be more results to come from here, is a lot of her bats have been roosting in rock cracks. So right here where this flag was, there was a bat roosting there during the day. And because this doesn't give you a lot for scale, here's another place where bats were roosting, and that's Shannon's hand. She's about my size. and there was one in those tiny, small cracks. So we've been really interested to learn about these cracks and these places where they're spending their days, because we really don't know a lot about where bats are spending the day. So this is what some of them look like while they're roosted in those cracks. The species she's focused on here is the little brown myotis. They're a very small bat, and it's a bat species that's been really negatively affected by white nose syndrome. So we're trying to get some good information about them. Through all of this work, through Shannon's work specifically, first we're going to try to provide some guidance about the kinds of places, the kinds of forests that bats like to live and bats like to forage. So we're thinking about characteristics of those trees that we can share with the managers. And then really for all of this work, we're just trying to get reliable information out to those that make the decisions and do the work on the ground. And with that, I will leave you with questions.